CD56 is a cell adhesion molecule that is normally expressed on natural killer cells and certain types of neuroendocrine cells and tumors. Malignant mesothelioma is a type of cancer that develops from the mesothelial cells that line the pleura, outer lining of the lungs, peritoneum, abdominal cavity, pericardium, sac around the heart, or tunica vaginalis, sac around the testes. Most cases of mesothelioma are caused by exposure to asbestos. Mesothelioma tumors are usually classified based on their expression of CD56. Epithelioid mesothelioma expresses CD56 in about 40 to 60% of cases, while sarcomatoid mesothelioma is generally CD56 negative in over 90% of cases. Biphasic mesothelioma shows mixed epithelioid and sarcomatoid patterns and can be either CD56 positive or negative depending on the specific tumor components. CD56 negative mesothelioma has been associated with more advanced disease stage and poorer prognosis compared to CD56 positive tumors. Several clinical trials have explored potential treatments for CD56 negative mesothelioma specifically, given its more aggressive behavior and limited standard treatment options. Here are some notable examples. A phase 2 trial tested the antibody-drug conjugate rovalbutamab, Rova-T, in patients with advanced CD56 negative malignant pleural mesothelioma whose disease had progressed after at least one prior chemotherapy. Rova-T targets delta-like protein 3, DLL3, which is overexpressed in many sarcomatoid and CD56 negative mesotheliomas. The study found Rova-T had an objective response rate of 15% and disease control rate of 52% in this population, suggesting it may provide a new targeted treatment option. Another phase 2 trial looked at nintedanib, an oral angiokinase inhibitor, in CD56 negative unresectable malignant pleural mesothelioma. Out of 27 enrolled patients, the disease control rate was 63% and median progression-free survival was 4.2 months, demonstrating modest clinical benefit from this anti-angiogenic therapy. Immunotherapy with the anti-PD-1 antibody pembrolizumab has also been explored specifically for CD56 negative mesothelioma in a phase 2 basket trial. Although response rates were lower than for other tumor types, some patients did experience durable responses, suggesting CD56 negative mesothelioma may still be sensitive to checkpoint inhibition in certain cases. Larger studies are still needed. A phase I/2 study evaluated the combination of pemetrexed and carboplatin with the anti-CTLA-4 antibody tremelimumab as first-line therapy for advanced CD56 negative mesothelioma. The objective response rate was 55.6%, median progression-free survival was 8 months, and median overall survival was 25.4 months, favorable outcomes compared to standard chemotherapy alone. This points to potential benefit from adding immunotherapy. Adoptive cell therapy using tumor-infiltrating lymphocytes, TILs, is another immunotherapy approach under investigation. One ongoing phase I clinical trial is assessing the safety and efficacy of autologous TIL infusion in patients with CD56 negative pleural mesothelioma. Preliminary results have shown the treatment to be well tolerated with promising response rates seen. While CD56 negative mesothelioma carries a poor prognosis, several clinical trials demonstrate that targeted therapies like Rova-T, anti-angiogenic drugs, immune checkpoint inhibitors, and cellular therapies may provide clinical benefit for this challenging subtype. Larger, well-designed studies are still needed, but ongoing research offers hope that more effective treatments can be developed to improve outcomes for patients with CD56 negative disease. Combination modalities incorporating different drug classes appear especially promising.